The first question was about showing a human heart and showing the pressures and also showing what would happen in this circuit. Important to show the pressures involved and the different pressures in the two circuits. So here's the right ventricle, the right atrium. Blood would be coming in here, being pumped by the atrium into the ventricle, and much thinner wall here on the right ventricle than the left ventricle. And so blood would be moving out, but at a relatively low pressure. Here's 25 millimeters of mercury might be reasonable. Heading out to the lung, it's a short distance, and you don't want high pressures in the lung because that would burst capillaries. You'd accumulate fluid and blood in the lung. And here is the left ventricle, left atrium, much thicker here, so it generates much more force. And so blood is going out at maybe 150, maybe 6 times higher pressures here out to the body. Has to go further and often a long distance, and so you need high pressures to force it there. What would happen if there was a hole in the septum? So here's our hole. Well, the pressures are a lot higher on this side, in the left ventricle, and so we're going to send more blood through the hole, and rather than out here, we're going to send some of our blood, maybe even a lot of the blood, into this much lower pressure right ventricle. Of course, these valves are going to be closed, and so that blood is going to go through the hole, and it's going to increase the pressure here. So instead of being 25 millimeters of mercury, depending on the size of the hole, it might be 50 or 70 millimeters of mercury. So we're going to send more blood under higher pressure. Some of this blood under higher pressure is going to the lungs. That's bad news. The lungs can't handle that, either the pressure or that capacity. We're going to send less blood out to the system, to the rest of the body, and so we're delivering less blood to the body and damaging the lungs. And of course we get some mixing of oxygenated and deoxygenated blood here, but no mixing here. The pressure's not strong enough to move any of the blood that way. I wanted to show you a couple others. This was just a really beautiful one, much more attractive than I could draw or than I intended, so I'm hoping this person did it because they were enjoying the exercise. But this is more of what a human heart really looks like with the blood vessels really nicely drawn. And here you can really see in this one how much thicker the ventricle is. How much thicker the left ventricular wall is, the part that is sending blood out to the system. I also wanted to show you one that's very straightforward and simple. This one shows the blood moving instead of having to explain it. Did leave out these valves. Make sure you include those valves on an exam. Pressures are shown here correctly. And so this person went so far as to show about the leakages, did not point out something I would want on the exam, which is that we're going to get higher pressure here and we're going to get lower pressure here. The next question was to show a capillary with the endothelial cells, including the basement membrane around the outside, and then showing how pressure helps to move water with nutrients out. Why don't large proteins and red blood cells leave? And then also the lymph vessels and what would happen if lymph vessels were blocked, which can happen. There's a parasite that does that. It's actually a nematode parasite. So here is a capillary. Well, here is an arterial leading to a capillary. Here is the beginning of the capillary. And we see water coming out under pressure, and it's carrying with it the molecules. So zooming in here, here's a cross-section through a capillary showing the capillary endothelial cells. I think this dash is supposed to indicate the basement membrane around the outside. And so there'd be basement membrane here. There are pores or gaps between the cells, and so valuable molecules, glucose, water, other small molecules are coming out, but there's this mesh here of the basement membrane, and so proteins don't get out. I think that's the this red here is proteins, and of course red blood cells can't get out. Truly in size in a capillary, a red blood cell would almost fill it, and then the fluid would be nearby or around it. So pressure is moving most of this out. There's most of that out. There's a little bit of diffusion as well, and then at the other end, there's water coming back in, carrying also some glucose, unused glucose, and so on. But it would be carrying some CO2 with it and other waste products to move away. Some of those wastes, of course, will also wind up in the lymph. There's more water that comes out than water returns because, remember, the proteins, the osmotic pressure is bringing some water back. But there's always a net loss of water, or on average, there's a net loss of water, and so that water has to go to the lymphatic system to get back and empty into the heart. So if we block this, if we have some parasite in there and block it, I think this zigzag is our parasite in there, then we don't move that water, so we have less fluid coming back. This person showed the lymph vessel going right back into this vein here. It actually would be an entire lymphatic system that they're just shortening it here. And the entire lymphatic system would bring the lymph back to empty into a vessel near the heart. What we'd wind up with is less water coming back. We'd have more blood cells, more protein, less water coming back, and so that would also cause a change in osmolarity. Here's just another example. This one also shows the the muscles around here, the precapillary sphincters that can close one of these off when you don't need blood going to that region. Here's the basement membrane and material coming out through the gaps in the holes. And then excess fluid going in here, in this case excess fluid not recovered, and tissue swelling would be another consequence. So here also it says cells swell, but it would be tissue swelling. So tissue swelling would be another consequence. This is edema, and that's what happens when lymphatic vessels are not draining properly. You get fluid accumulation in the tissues. The next one was to think about the white blood cells, leukocytes or white blood cells. They are immune system cells. They're about 1 in every 1,000 cells in your blood. And there's more than one cell type. In this case, I'm talking probably about macrophages, which are attracted to areas of damaged cells. And what I wanted you to show me was damaged cells releasing molecules. Those would be damage-associated molecular patterns, or DAMPs. There's also pathogen-associated molecular patterns, which are coming from bacteria or viruses or parasites. So we get these DAMPs. So here is a DAMP, gets into the blood, and here is a white blood cell going by, and it binds to it. What I wanted you to show was a molecule that could cause release of vesicles that might have enzymes or other molecules that will attack and kill parasites, disease, damaged cells, whatever, and break down that tissue. Of course, we wouldn't want it to happen in the red blood cell. We'd actually want this to happen in sequence, where our white blood cell moved out into this area and then released its enzymes and whatever. And I also wanted you to show this signal causing a pseudopod to develop. And that would be, that is actually what happens. A white blood cell is just happily moving along in here, but when it binds to some protein that signals damage nearby, it becomes amoeboid, squeezes out through the gaps between the cells, and then it grows pseudopods to move toward the areas of damage. It's following a trail of this. And so what we'd want is wherever that binds, we activate this pathway. Here's cyclic AMP being activated by a G protein in this case. And in this pathway, the G protein is activating actin assembly, a microtubule assembly protein. And so in the region where this binding is, that's where the microtubules would be assembling, and they'd be moving, in this case not in this direction, because this message won't get to the far side of the cell. This person just didn't have enough room to show it. The message would be here, and our actin microfilament assembly protein would be growing this way. So the pseudopod would be going in the direction of highest concentration of our messenger. And here's just another one showing something similar. I included this one because this one was using, here is actin, And then here's some myosin tethered here, and the myosin is pushing the actin to form the pseudopod. Again, we'd want that activation to happen only near where binding is taking place so that the pseudopod is moving our cell toward the area of damage. The next one, cyanoatrial node of a person expresses fewer potassium channels. Now this was open-ended. You could decide it was fewer leak channels. So it could be leak channels, could be voltage-gated channels, and different people answered it different ways. If it's leak channels, then the heart rate would increase because with fewer leak channels, remember potassium leaves the cell. So if you have fewer of them, you'd still have some potassium leaving, but you're trapping some potassium inside the cell. You're going to depolarize it faster from -70 toward -45. It's actually different in a heart cell, but anyway, you're going to get to threshold sooner. You're going to have a faster heart rate because you've got a more positive resting potential, more frequent action potentials. If it was more voltage-gated potassium channels, then you would have the action potential, which is very long in the heart, and then comes down. Here's where voltage-gated potassium channels are important. If you had fewer of them, you'd have a slower pace, and you'd actually slow down contractions. Finally, number 5, a person has damage to specialized cells that conduct the action potential from the AV node, atrioventricular node, through the ventricle. And what would that do to atrial contractions, ventricular contractions, and how would this person perform? Here's an answer assuming the damage allowed slower transmission of the action potential. Again, you had choices as to how to answer this one. If they're slower than normal, in all cases, atrial contractions would continue as normal. But if the damage slows down passage, then you delay ventricular contraction. They're going to be slowed because it takes the action potential longer to reach it. And this person is going to have trouble exercising because it takes them longer to get oxygenated blood to the limbs. So that person is going to have trouble. Here's another one, assuming the damage allowed faster transmission of the action potential, and you could do it a third way, which is no transmission. So assuming faster transmission of the action potentials, then the not going to completely empty the, it says ventricles, I think they mean atria here, because the atria are pumping normally, no change to the SA node, so atrial contractions are normal. So the atria are, let me think, what is this? So if it's faster transmission, then the atria would not completely, the atria would not completely empty into ventricles before ventricular contractions. And because you got a faster transmission of the action potential, the ventricular contraction is sooner. So timing of atrial contractions would be the same. So you'd have premature ventricular contraction, wouldn't be filled completely, so you're delivering less blood to the tissues and to the lungs. So you're not going to get enough oxygenated blood to tissues, going to fatigue easily. Heart rate would increase to try and match that. That's correct, to try and correct for that, but the ventricles are still not filling correctly. You could actually, if it's bad enough, this sort of damage can cause heart failure. There's also a suggestion it would cause an arrhythmia, and that's also another good guess.